In this video, I show you how to visualize your own data on the UCSC Genome Browser using custom tracks. I use the example of ChIP-seq files for big datasets. I explain file formats and show you the steps of how to upload your data via your own free web server space. I also show you how to create your own colourful genomic annotations so that you can generate attractive and professional scale presentations. A key feature of the UCSC Genome Browser is that you can visualize your own genomic data and create your own annotations, which will be presented dynamically and at scale when you use the browser. The browser accepts many types of data in a wide variety of formats, but you do need to take care to follow these formats correctly. A key thing to point out from the start is that the UCSC Genome Browser is not a data repository. If you upload data directly to the browser, it will only be stored temporarily and will often disappear the next time you come back. Also, the browser is a public facility. While your data analysis should be secure in your account, you need to be careful when browsing patient data or commercially sensitive data using public tools. So, before you start, consider what data you have, how large is it, and what restrictions might there be on analysing it off-site. Your data will only be visible to you or to anyone you decide to share your browser session ID with. If your data is stored on a third-party server, which I'll cover in a minute, you can save a session at the UCSC Genome Browser that will remember all of your settings and all the links to your data. You can return to the session at any time in the future, share it with collaborators, and even export it to a different browser mirror site. This is a fundamental productivity advantage of the UCSC Genome Browser. There is also the option to go a step further and establish a track hub, which is a more stable option for sharing custom annotations with others. Track hubs are the preferred approach if you have a lot of data tracks to view and share. Look out for our tutorials explaining UCSC browser sessions and track hubs on this channel. In general, the facility that you've been using to generate genome scale data will also have tools to generate browser compatible files. If you've used the UCSC Genome Browser before, you're likely to have browsed public data tracks similar to your data type, so you will have a very good idea of what format of file you need to create and how you want to display it. There is an in-depth guidance on browser file formats on the UCSC Genome Browser Users guide, and I've linked to this in the description. In this tutorial, I'm going to show you how to visualize ChIP-seq data, which displays the genomic locations of transcription factor binding or histone modifications, for example. And I'll use this as an example of a big data format. I'll then show you how to create your own annotations of genomic features using the bed file format. From these examples, you'll get a feel for how to approach your own custom annotations. ChIP-seq analyses involve the cross-linking of proteins to the genomic locations in living cells. Cross-linked chromatin is then fragmented, and proteins or chemical modifications of interest are then isolated using a specific antibody. The pool of enriched genomic fragments is then isolated and sequenced in high throughput. Tens of millions of reads are typically aligned to a human genome, for example. The resulting binary alignment map, or BAM files, can then be viewed to show the enrichment of read alignments at specific genomic sequences where a factor of interest was located, but these are very hard to read on a browser. In order to generate a ChIP-seq track for a genome browser, we firstly segment the genome into small portions such as 10 or 50 bases. These are called bins, and we measure the number of reads that are aligned within each bin. A ChIP-seq track is essentially a graph of normalized read enrichment versus genomic position. This kind of graph of continuous data is called a wiggle or WIG track. You can use the same approach for displaying RNA-seq or any other genomic enrichment data. The size of whole genome ChIP-seq datasets is such that it's impractical for you to send the whole track file to the browser every time you make a query to what enrichment values there are at a specific genomic region. You can't send this whole file every time you zoom in, zoom out, pan left or right. So an index binary file format called bigwig is used. The advantage of this format is that only the portions of the file needed to display a particular region are transferred to the Genome Browser server. This greatly speeds up the display performance when working with large datasets. The caveat is that you need to place your bigwig file onto a web accessible server first, and only the portion of the file that is needed for the currently displayed chromosomal position is locally cached onto the UCSC server. So, in addition to creating browsable data files like bigwig, you need to upload them onto a file server, HTTP or FTP server, and then share a link to that file with the UCSC Genome Browser. So you're going to need a host site for your large data files. The information technology service at most institutions will be able to help you out with server space for your tracks and track hubs, but sometimes there are restrictions on how you can share files publicly from your institution. So if that proves to be difficult, you could use a commercial web space or a cloud storage provider, and I'll leave a link in the description to a list of hosting options that you could look at. There are some free web space providers, and one that I recommend is the CyVerse Discovery Environment, a publicly funded web browser platform based in the US and the UK that provides life scientists from around the world with powerful computational infrastructure to handle huge datasets and complex analyses. This does also include the free hosting of your data files for uses like displaying them on the UCSC Genome Browser. And this is a service we have used with success for several years. If you've not used the CyVerse Discovery Environment before, you firstly need to create a free CyVerse account, and a link to this site is provided in the description. Once you have done that and logged in, you will see a Data tab which opens up a file manager. Here you can create subfolders within your account and upload your files. The simple web upload may be slow depending on your needs, but there are several ways to upload data, and you can follow the guidance on the CyVerse page. Once uploaded, you can click on the three dots by a given file and select the Create View a Public Link option, and then you need to copy this hyperlink into a text file on your computer, and this you can refer to later on. Once you've uploaded and created links for each of your data tracks, you can leave CyVerse, although there are lots of apps on there that you may want to explore at another time. The next step is to create the track lines, lines of text which instruct the browser where to find your data, what to call it, and how to display it. There are many attributes that you can add into your track lines which give you a lot of control over the display of your data, and I've included a link to the track line options in the description. You need a track line for each data file that you have, which follows this format. So this is one line of text with no line breaks in it, and a single space between each attribute. The first one defines the track type, that's essential, and what you put there should be obvious, and again, guidance is in the user's guide. The name is what is shown on the left hand side panel of the browser data window, so you can control that. The description is the text displayed above your track. If you've chosen to show that, I don't use that functionality. The color is an option. The default is black. You can use red, green, blue or RGB codes to define any color for your data. And I should point out that Microsoft Office also uses RGB codes for colors. So you can easily pick any color in an Office program and find the RGB values there. So there's even a color picking tool where you can point at a color that you're trying to match and it will give you the RGB code. So this is very useful when trying to match colors in a presentation. A great functionality. Visibility is also optional and defines the display mode for your data track, where 0 equals hide, 1 is dense view, 2 is full view, 3 is pack, and 4 is squish. The numerical values, all the words can be used, and the default, if you don't set anything, is 1, which is dense. The link is essential, of course, and should be the full address of your web accessible file. So it needs to include, for example, http://, for example. HTTP links perform the best, although FTP links can also be used. You can assemble these track lines quite quickly in Microsoft Word. Each attribute is separated by a single space. Just make sure there's no hidden line breaks or extra spaces in your track line. When you copy paste into Microsoft Word, it can, for example, add line breaks that you didn't want. So here are my examples for this tutorial, and you can see I've got several ChIP-seq tracks here, and I've decided to use different colors for each based on how I want it to display. So now you're ready to upload your data to the UCSC Genome Browser. Click the button Add or Manage Custom Tracks, and that's at the bottom of the browser window in the center. So click on that, and then you copy paste your track lines into the box that says Paste URLs or Data, and then click Submit. The track should upload quite quickly, and you'll see a table of all your custom tracks. If you've got any errors, then follow the description that the browser's given you as to where your error is, and often you've got an extra space or line break that you didn't intend to use. So now you can go view your files in the Genome Browser. In this tutorial, we're just going to move on now to creating an annotation of genomic features. So for this, you need to create a short text file in the Browser Extensible Data, or BED, BED format. In this example, I have the sequences of some gene regulatory elements for the TAL1 gene, and I'd like to annotate these on the browser. So then I can compare the ChIP-seq profiles of the transcription factors that I've just uploaded and see how they match up to these known gene regulatory elements. I can use the BLAT tool within the Genome Browser to quickly find the genomic coordinates of these sequences. So this is like BLAST, it's a very quick alignment tool, and you can find it here in the top menu. You need to enter your data sequences in the FASTA format and also choose the correct genome build that you want the coordinates for. The FASTA format's very simple, a forward arrow followed by a name, no spaces in the name, then a line break, and then just the text of your sequence. The results are produced very quickly, and you can copy paste these coordinates into a text editor to then create your BED file. So here is the output from my search that I've just shown you. This is one of the BED formats that produces coloured genome annotations. Note that there are several different kinds of BED formats, and again you can follow the user's guide for lots of different examples. So here I want to create boxes on the Genome Browser drawn to scale, and I want the boxes to be different colours for the different kinds of elements that I'm annotating, and I obviously want the names next to them. So this is the correct format for that. So the first row, which is optional, specifies the location which the browser should open at. This is useful for directing to TAL1, the TAL1 locus in this example. The second row tells the browser to hide everything else that you don't want to see. I generally don't use this line. I'd like to control what's in the browser myself. The third row has attributes for the name, description, and track visibility settings, as I've just discussed in the track line section. There's also an instruction to use item RGB color codes, which is exactly what I want in this case. So rows 4 onwards contain the data lines. So you have the genomic coordinates in columns 1 to 3, followed by the item name in column 4. The item name, don't put any spaces in there, use underscores if you've got multiple words. You have the option to define the direction of your annotation if that's important for your particular features. And then the second set of coordinates define the region which should be drawn as a full box. In my case, I want a solid rectangle, a full box, for all of my gene regulatory elements. So I copy my coordinates into these columns again. The last column has the red, green and blue values, which range from 0 to 255, and each of those is separated by commas. Each column in these data lines is separated by a tab. Be careful not to have any spaces in the data lines. The browser will reject your bed file if you do. So here are the genomic features for my example in bed format. So I've got the results from the alignment that I showed you, and I've added in a few more. I direct the browser to the TAL1 locus in row 1, and I've copied the item names and genomic coordinates into the data rows below. Note that the coordinates appear twice in this format. I've chosen red, which is 25500 for promoter elements. So 255 is the maximum red, no green, no blue. 255000 is red. And then I've chosen yellow and blue colors for enhancers and CTCF elements in this example. These bed files take a few minutes to put together, but they do give you complete control over your annotations, which of course will be to scale in your final presentations. So this can be really powerful if you need to annotate regulatory elements, primers, all sorts of things like that. And just bear in mind that if you didn't take the time to do this as a bed file now for the Genome Browser, you probably would be drawing a figure, something like this later on in PowerPoint or some other graphical tool, and that wouldn't be to scale. So it really makes sense for some projects to create these bed files. You can copy paste this bed formatted text directly into the Genome Browser custom track page as before, but it's actually best to save it as a plain text file, which you can use later on and refer back to later on your computer. And then you can upload this file directly into the Add Custom Tracks page. Be careful if you've used a program like Microsoft Word or Excel to create your bed files. If you've got lots of tracks, you might be doing all of this in Excel. Be sure to save your file as a plain text file, or if you're in Excel, save it as a tab delimited text file. Then open up your output file in a plain text editor and just check the formatting. Make sure there are no extra line breaks or spaces. And in the case of Excel, those quotation marks in the top lines will actually get doubled, and again the bed format will be rejected. So just edit out any Microsoft helpful additions. I've uploaded my gene annotation now, and I shall briefly describe what I see in this session. So I've arranged my tracks by dragging and dropping them, and what I've done is put the transcription factor tracks on the top, the histone modification ChIP-seq tracks at the bottom, and in the middle I've got the gene annotation, and then I've got my annotation of gene regulatory elements from my bed file. So what do we see? So we can see firstly that the CTCF signal in that ChIP-seq profile is very high at known CTCF regulatory elements, which are shown in blue. The GATA1 and TAL1 transcription factors, which are shown in red, are co-binding at one of the TAL1 promoters and at this downstream enhancer element. The promoter associated histone modification I'm showing here in green is enriched at the promoters of the TAL1 and STIL genes. And the PDZ gene is not active in this cell type, so there's no modification at that. Finally, the enhancer associated mark shown in yellow is showing many gene regulatory elements are active in this cell type, so there's an awful lot going on. So if I was a researcher adding new information into the regulation of the TAL1 locus and adding new features, I could then overlay it with all this other information and get a leg up of a quick start as to what I'm looking at, where I am, and how to contextualize it with this other information. Very, very, very powerful use of the Genome Browser. So I hope this example has given you confidence to upload your own data into the UCSC Genome Browser, and to use the functionality that's available to you to make your presentations look clean and professional. I hope you found this tutorial to be useful. Do us a favour in return and click the like button, and leave a comment below to let us know what you liked, or what else you would like us to cover in the future. We have lots of other lectures, tutorials and interesting videos on the channel, so please browse our channel page and consider subscribing. Thanks for watching.